Good evening, everyone. I'll wait for 2 more minutes and then after that we can start the session. I hope I'm clearly audible. If yes, you can just write, someone can write yes or no in the chat box and then I'll start. Okay. Okay, so I welcome you all to this fourth problem solving session for the NPTEL course Cell Culture Technologies, which is taken by Professor Manak Das. I am Pushpangna and I am a PMRF scholar from IIT Kanpur. So today we'll be discussing some model questions based on which you also have to solve your current week 4 assignment. So if you have any doubt in answer to any question or any doubt in general, what you can do, you can drop your questions in the chat box, and if time permits, I'll take up those questions and we can discuss them after I have discussed these model questions. So the first question is, poly D lysine contains how many nitrogen atoms? So if someone knows the answer, they can write in the chat box and then I'll explain. So the correct answer is poly D lysine contains two nitrogen atoms. So in the last session itself, we discussed about what is poly D lysine So this is the structure of the poly D lysine compound. And as you can see here, it has two nitrogen atoms. So what actually is poly D lysine and why are we discussing about it in our cell culture class? So poly D lysine is a synthetic compound and it is used for providing adhesion in your cell culture substrate. By cell culture substrate, I mean your Petri plates or your T50, T25 flasks that we discussed in the previous lecture. So what happens is for adherent cell lines, you need some kind of matrix or some kind of support through which they can attach to your substrate, to like to your plate. So for that we use the synthetic compound that is poly D lysine. So it is actually a polymer of this unit. And what poly D lysine does, very briefly I'll recap again. So it has positive charge and the cell membrane or the DNA which is shown here in this figure, they have negative charge. So what will happen because of this electrostatic interaction? The negatively charged cells, because of their cell membrane or your negatively charged DNA, can go and get attached to poly D lysine, and that is how your cells can adhere to the glass surface. So here also in this figure, you can also see here it's given mitochondria, but you can imagine a cell which has negative charge all over its membrane. So it can now it can go and attach to the positively charged surface which has poly D lysine. So this is the purpose for which poly D lysine is used for cell attachment and adhesion in cell culture. So the next question is, which of the following is a synthetic extracellular matrix? Now there are four options: matrigel, collagen, laminin, or poly-L ornithine. So if someone knows the answer, they can mention in the chat box. And this thing has also been discussed extensively in one of the week 4 lectures. I think it's the first lecture by Manak sir. So here we are talking about synthetic ECM, synthetic extracellular matrix. So a synthetic extracellular matrix out of all these is poly-L ornithine. Matrigel is not synthetic. We discussed about matrigel in the previous lecture. Like we discussed extensively about what is matrigel and what are its components. So it is extracted from a mouse sarcoma. If you want more details, you can visit the previous lecture. I will provide the link at the end of this session in the chat box. So you can look up matrigel there. Collagen and laminin are not synthetic. They are produced naturally. They are part of naturally occurring ECM. While poly-L ornithine is a synthetic matrix. So if you remember, so this is poly-L ornithine. And this is in laboratory, you will either find it in this kind of powder or this kind of solution form. But this is the basic composition of polyl ornithine. It is usually found in a hydrobromide form, but this goes away later. So if we talk about the different kinds of ECM, then they can either be synthetic or natural. Now again, your synthetic and natural ECMs can be of different types. Your natural ECM can be collagen or laminin, but your synthetic ECM is poly-L-ornithine, and there was one more synthetic ECM which was mentioned in the NPTEL lecture, that is DETA silane. So this information is purely from the lecture which has been uploaded. So natural can be collagen, laminin, even gelatin for that matter is a natural ECM. But your synthetic ECM is polyl-ornithine, DETA silane, and also your poly-D-lysine is not particularly an ECM, it is just a synthetic compound which helps in adhesion. So next, moving on to the next question. Gelatin is a hydrolyzed form of collagen, elastin, laminin or fibronectin. So gelatin basically is a hydrolyzed form of collagen. So when I say gelatin, what comes up in your mind? So if I say gelatin, you will usually think of some confectionery items or something like this kind of candy or like some gel kind of substance. So it's true, basically gelatin is used in confectionery and in making these kind of items. But gelatin basically is found in this kind of a powdered form. And if you look at the composition of gelatin, then it is, since it's a protein, it's rich in amino acids. As you can see in this profile, it has different kinds of amino acids: alanine, arginine, glutamic acid, glycine, and so on. But gelatin is actually formed by partial denaturation of this collagen triple helix. So if you look at the structure of collagen, it is actually triple helical, and once you do the partial denaturation or once you heat collagen in very simple terms, then this ordered protein backbone breaks down and then gelatin is formed. So basically, if you look at the structure of collagen, it is a triple helix like this. As you can see, there are three helices, and when collagen is present in our when collagen is present as an ECM, it provides structural support. So basically, the chemical composition of collagen and gelatin are same, but on one hand we have collagen which is providing structural support, but on the other hand we have gelatin which has a jelly-like consistency. So why is it resulting in this kind of difference when we have the same chemical composition? So when you take collagen and when you do acid and then base hydrolysis, then what happens? This highly ordered triple helix of collagen breaks down and it forms a disordered structure, which is our gelatin. So what happens is when we do partial denaturation or when we heat collagen, then what will happen? Like these water, once the gelatin cools, once this cools down, Then water molecules get trapped. As you can see, these are the water molecules, and these water molecules get trapped in the structure, and this leads to formation of like when gelatin solidifies from its solution form, and it forms a gel. So what the jelly kind of consistency that you see is because these water molecules have been trapped inside your solution form of gelatin, and that is why you see the jelly-like consistency. But gelatin essentially is a hydrolyzed form of collagen. Now moving on to the next question. Atomic force microscopy helps us to analyze. First is chemical constituents in the bulk of material. Second is topography of the surface. Third is concentration of material. Fourth is electrical stimulation of cells. So if you know the answer, you can mention in the chat box. So atomic force microscopy, or AFM as we call it, helps us to know the topography of the surface. So I'll discuss very briefly what is AFM and how does it work. So this is a schematic diagram of a very basic setup of ASM. So this is the setup, and this is how a atomic force microscope looks like. So what happens is, what your atomic force microscope will do. So you have this cantilever which has its tip. So here what will happen, suppose you do not have an even surface, like you do not have an even surface like this. You have somewhat of an undulating surface. Just a second. So you have a surface which is like this, which is undulating and which is uneven. Now, but you do not know what kind of a surface this is. So basically, you do not know the topography of the surface. So here, what will happen? So your cantilever has this very sharp and small tip, and it will scan the area over the surface. So take into account that whenever the cantilever will be contacting the surface, for example, if the cantilever is contacting the surface here, and if the cantilever is contacting the surface here, what will happen is your cantilever will bend in accordance with what is the depression or what is the topography of the surface. And as a result of which this cantilever is connected to this laser, as you can see. So as a result of which the amount of laser light which is being reflected in this photodiode will differ. So here what will happen, according to the topography of your surface, the reflection of your laser light will change. So from here, depending on what kind of reflection and what is the amount of reflection of your incoming laser light, you can indirectly understand what is the topography of the surface. So this height of the cantilever, this cantilever and its height will also change in response to your topography or the height of your surface. So this is how, uh, this is the basic principle of the atomic force microscope. However, as the name suggests, atomic force, there is no nuclear force or anything like that involved. And this photodiode is the main detector which measures or which takes the output of how much laser is being reflected depending on the kind of sample surface which is there. If we look at the basic, if we look at how a basic AFM microscope looks like, so this is where. So in modern day microscopes, we usually have this high resolution video cameras which are there so that you can record whatever you're seeing through your objective. And then we have a Z stage. Z stage meaning it can move in all the three directions, X, Y, and Z. Then we have a liver force sensor. Please mute yourself if you have any doubts or if you want to convey something, you can always put it in the chat box. Okay. Then we have this XYZ piezo scanner. So I forgot to mention this. So this PZT is actually a piezoelectric translation stage. So I'll just write it here. So what do we mean by piezoelectric? Basically, piezo means pressure. So something that is being generated by pressure. So here what is happening? You are measuring the kind of pressure that your cantilever is experiencing, and that is why this is a piezo electric method or piezoelectric stage. So moving on to the next question. X-ray photoelectron spectroscopy is used to analyze. First option, chemical constituents at the surface of material. Second, topography of the surface. Third, chemical constituents in the bulk of material. Fourth, topography of the bulk. So you can answer if you know in the chat box. So X-ray photoelectron spectroscopy is used to analyze chemical constituents at the surface of the material. So X-ray photoelectron spectroscopy or XPS, it has another name which is electron spectroscopy for chemical analysis, or ESCA. So this X-ray photoelectron spectroscopy is a very widely used technique, and it is used to analyze what is the chemical constituent at the surface of any material, and it can also analyze the composition and the different bonding states because it's using X-ray on the sample surface. So this is the basic setup of our XPS. So what are the information provided by X-ray photoelectron spectroscopy? So as I mentioned earlier, First will be the chemical analysis at the surface. When I say chemical analysis at the surface, then uh, composition, composition of the elements which will be constituting your sample surface, and then through XPS you can also know the chemical bonding state. So how does XPS work? So with any spectroscopy or any microscopy, you need to have a source. So here your photon source can either be an X-ray tube, a UV lamp, or a synchrotron. So from here your photons are coming and then they are hitting the sample. Once they hit the sample, once your photoelectrons are emitted from your sample. So these are the photoelectrons. I'm not going into a lot of detail. I'm just like telling you the basics of the technique so that you can know what is the relevance and connection to cell culture rather than going all in the physics of it. Because the important thing to know is why are we talking about all these techniques in cell culture and what is the basic working principle, that's all. So once you have your sample and your photons coming from any of either of these three sources hit your sample, then they will emit your photoelectrons, and from this emitted photoelectron you can analyze the change in bond energy or you can see the chemical shift, and through this through this information with the help of this energy analyzer which will be collected by the detector, you can know all this information like what is the chemical constituent at the surface, what is the compositions of the elements which are constituting the sample, and what are the different chemical bonding states. So moving on to the next question. Contact angle on the surface is indicative of what property, and what is the instrument using which it is measured? So is contact angle a measure of hydrophilicity or hydrophobicity? And how is it measured? Using osmometer or goniometer? Yeah, so people have answered. So contact angle is a measure indicative of hydrophilicity and it is measured using goniometer. So the correct answer is C. Now here, if you look at this leaf surface, then you can see that this is a beautiful water drop which has just fallen on it, but it has not spread on the entire leaf. Rather, it has formed this circular spherical shape, and you can see the subsequent water drops also. Here also, as you can see at the tip of this leaf, this water drop is there, and still it is completely spherical. So, what is the reason that when these water drops are falling on the leaf surface, they are not spreading, like which is the normal property of water. Rather, they are forming this spheric sphere, and they are trying to minimize their contact with the surface. So basically, water will try to minimize its contact with the surface because this surface is actually hydrophobic. Hydrophobic means, so hydro means water, phobic means repellent. So basically, this surface is repelling water. It's not allowing water to like it's not allowing water to come in contact with it, and that is why it's forming a sphere so that it can have minimum contact with the surface and minimum angle with the surface. So this you must have observed this phenomena in many leaves or in many natural ways. Now, if you look at, so depending on the nature of the surface, whether the surface is hydrophobic, hydrophilic, or what, you will have water behaving different with them. So, so suppose if you have a hydrophilic surface, then the contact angle of the water with the surface will be different, as you can see here. If you have a hydrophobic surface, then your contact angle will be different. Here the contact angle is less than 90 degree. It's between 0 to 90 degree. And when the nature of the surface is hydrophobic, then the contact angle is different. As you can see, it's between 90 degree to 150 degree. If the surface is super hydrophobic, then the contact angle will be more than 150 degree, and this was the case here in this leaf. The surface was highly water repellent, that is, it was super hydrophobic, and that is why it had a huge contact angle, like it had a high contact angle, so that this droplet itself, this water droplet, can have as minimal interaction with the surface as possible. So now, what actually is the contact angle? So as you can see here, is the angle which this water droplet or this water will make with the surface. So this theta here is the contact angle. Now, for our laboratory purposes, why do we want to measure contact angle? Because what happens is, suppose you have your cell culture dish or vessel or a flask, whatever you're working with, and suppose you have added your ECM, or you have coated your plates with poly D lysine or matrigel or whatever means of adhesion you have. Now, when we perform these kind of coatings with our natural ECM, synthetic ECM, polylysine, matrigel, polyornithine, then what we do, we have a specific concentration. We do not just randomly go and pour the solution no matter whatever its concentration is. So there's a reason why we need that concentration to be specific, and there's a reason why we do not need, suppose that specific concentration is X. So we do not want the concentration of the ECM solution that we have to be very less than X, and we also do not want the concentration to be very more than X. And there's a reason for that. Why? Because suppose the concentration that you need is X. Then what will happen? You have coated your plates with this X concentration of the ECM solution. Then your cells will attach normally to your plates, and you'll see that they are growing and dividing just fine. However, suppose if you have a concentration of the ECM which is suppose 100x. So you have coated your plates with 100x the concentration of what was the concentration which was recommended. Then what will happen is you will think more ECM, more adhesion, the cells can now attach more strongly to the surface. But that is not the case. Why? It's because if you go on increasing the concentration of your ECM or concentration of poly D lysine, then your surface will tend to become hydrophobic. And as a result, your cells will not be able to attach to your surface. And that is the reason why you need to know that with increasing or decreasing concentration of your polylysine or your ECM, the nature of your surface is going to change. And because of this change in the nature of the surface, your cell attachment is going to happen differently, and this will start a chain reaction, and that is going to hamper your entire long experiment. Hence, it is important to either optimize the concentration of the ECM which you are going to use if it's not optimized in your lab before, or if it's already optimized, not to exceed it too much. So, as we were talking about contact angle, so this is why we are using this term, this phenomena, contact angle and goniometer in our cell culture technologies class, because suppose you do not know why is the reason that your cells are not attaching to your ECM even when you are using, according to you are using a good concentration, or this is the concentration which should be used. So you can measure the contact angle which your cells are forming with your ECM or with your surface, and through that you can know that okay, this contact angle is not good enough for the cells to attach properly or for the cells to grow. So this is where this contact is used, this phenomena is used. So basically, since we are talking about contact angles, a contact angle is measured using a goniometer. So this is the basic working principle of a goniometer. As you can see, so here I'll explain the setup very briefly. So we have our lens here. So the lens is connected to this camera, which in turn is connected to PC, where you can see. So this is the camera image which is coming on your PC. These two. So this lens is connected to your camera, and then what will happen? So you have your syringe pump here. So this is the syringe pump, and then this is your test surface. So here, what the syringe pump will do? The syringe pump will dispense a specific volume of the liquid on your test surface, and once it does that, you this camera and this lens will record that movie, and once it is done we can analyze what is the what is the contact angle that that specific liquid or that specific liquid is making with the test surface. So here, as you can see, so this is like the tip of your syringe pump, and this is your test surface, and this is the volume which has this is the liquid which has been dispensed. So as you can see here, like since you have recorded the movie already, you can see when the volume, you can play it in reverse order, and you can see when the volume is decreasing, then what is happening, and when the volume is increasing, when the pump has dispensed the liquid on the test surface. So there's a video which I can show you, so you can appreciate how a goniometer works. So this is a YouTube video. The link is given in the presentation, which I'll upload. So this is the syringe pump that I was talking about. I hope the video is visible. Okay, it's visible. So this is the syringe pump that I was talking about. So this is the analysis software, and this is the camera. So this is the tip of the syringe pump. And once your goniometer is switched on, then here you can see that this tip is visible. Now you can see this is this specific liquid which is present, and this is the test surface which we are going to test. So now you will see the liquid being dispensed onto the test surface. So you can appreciate that as the liquid is being dispensed onto the test surface, there is this high-end camera which is recording this entire thing, and even when the volume of the liquid is very low, it may be somewhere around 10 to 20 microliters only, then also this is being recorded. And just through 10 to 20 microliters volume of that liquid and that small test surface, you can predict the contact angle and the kind of relation that the liquid is going to have with the test surface. So now the syringe pump has dispensed the liquid on the test surface, and we have the video which has been recorded. So now that you have recorded your experiment, the next step is to analyze uh, like analyze what kind of contact angle is being made between the liquid and the test surface. So as you can see here, now you can move it forward and backward, and you can see what is the contact angle and the diameter of the droplet which is being formed. So as you can see, using this, you can see the contact angles, this blue line and the red line. Now you can easily see the contact angle which the liquid is making with the test surface. So basically this is how a goniometer works. Okay. So moving on to the next question. Concavalin A is a carbohydrate binding protein, a protein binding carbohydrate, protein binding lipid, or a lipid binding carbohydrate. Yeah, so concavalin A is a carbohydrate binding protein. So actually concavalin A belongs to a specific group of proteins which are called lecithins, and they specifically bind to carbohydrates. So this specific compound concavalin A actually binds to mannose. So it binds to mannose residues, and it can lead, like the presence of concavalin A can lead to different kinds of uh, reactions in the body or in the test subject, which it can trigger a kind of immune response, it can trigger cytokine storm, and so on. So here, as you can see, here's the structure of concavelin A, which I have taken from a research paper whose link is given. If you're interested, you can go and check it out. And here they have made some peptides which have mannose residues and they can bind to concavelin A. So moving to the next question. The next question is retina contains rod and cone cells. So what do rod detects and what do cones detect? Yeah, so rods detect the intensity of light and cones detect the color of light. So this is the basic structure of the rod and cone cells which are found in our eyes. So both these rods and cones have something which are called photoreceptors, and this is how they detect the light, whether it's intensity or color. And another important thing to note in these kinds of cells is that they have an extensive, like they have extensive ion channels and ion pumps through which they constantly and actively transport sodium and potassium ions. So as you can see here in the structure, it has a different sodium potassium pump, then it has non-gated potassium channel, then it has active transport sodium channels and so on. From this question, from this uh, brief discussion, I move on to the next question, and you'll find a hint for the answer here. So rods and cones are electrically active, sound sensors, pressure sensors, or electrically inactive. So just in the previous description, I mentioned that the rods and cones have extensive like these ion channels and they constantly transport sodium and potassium ions in and out. Yeah, so rods and cones are electrically active. Now, so an electric pulse or an electric impulse is generated when the sodium and potassium ions move in and out of the cell. So basically, this is a detailed diagram where you can see, where you can go through how the cones or rods are becoming depolarized or polarized and how their electrical activation is taking place. So this is the last question. The powdered collagenase should be stored at all times desiccated at 20 degrees Celsius, 0 degrees Celsius, minus 4 degrees Celsius, or minus 20 degrees Celsius. So the correct option is minus 20 degrees Celsius. Collagenase is an enzyme, and as most of the enzymes, it will get denatured if it is stored at a low temperature, higher temperature. So that is why collagenase, especially powdered collagenase, is stored at a temperature which is sub-zero, and that is at minus 20 degrees Celsius. So these were the set of questions for today. If you have any doubts or if you want to ask any question, you can put it in the chat box. So maybe we can discuss that for another 2-3 minutes and then I'll stop the session. For your reference, I'm sending you the link of the playlist, the YouTube playlist. so that if you want to look at previous lectures or previous videos, you can do that. So I've sent the link of the playlist. There was a question at the beginning of the session about the pattern of the enzyme for this course. Sorry, pattern of the exam for this course. So I'm just here to solve, like, to help you guide in solving your assignments. I do not know very well about what will be the pattern of the exam, whether it will be subjective or objective. What you can do, if you have any doubts, you can post it in the discussion channel, or what you can do, you can write a mail to the NPTEL team. Answer to the last question is minus 20 degrees Celsius. Polyl ornithine and ornithine mean same. No, they are not the same. So basically, ornithine is synthesized from glutamate, I think, in the body, like basically in that urea cycle. But polyl ornithine, so polyl ornithine, as it says poly, so it's just not. So this is polyl ornithine. So as it says poly, that itself means it's going to have many units, many repeat units of the same thing. So this is the basic structure of ornithine, and then it's going to have different, like, same repeat units. So that is why N is written. But polyl ornithine is not found naturally; it is synthesized. So it is a synthetic compound. L represents chirality. So, like, you must have read about chirality, L in D forms. So it is the L form. So notes for this, I do not know if we are allowed to give notes. You can, so I posted uh, the YouTube playlist, the link for the YouTube playlist. If you have any doubts, you can, like, go through the videos again. Again, if you have some doubts, then what you can do, you can always join these NPTEL sessions. It is not just only me who is taking the sessions. There are two more sessions scheduled on Tuesday, so you can so you can join those sessions as well, and then you can always go through the videos. They will give PDF notes. Yeah, so about notes and about the pattern of the exam, you will have to write a mail to the NPTEL team, because we are scholars from IIT, and, like, we can only aid you in solving the assignment questions. We are not dealing with the NPTEL notes or the NPTEL exam. We are here to help you solve the assignments which are provided every week.
Ma'am, one doubt, ma'am.
You can write it in the chat box. I'm going through like, questions one by one. Both can be used to coat lamin. Both means polyol ornithine and ornithine. So no, just ornithine cannot be used for coating, and that is because, so you need a So you need a matrix which is going to be formed only through a polymer, only by a polymer, and that is why we use either polylysine or polyornithine. Just you cannot obtain a matrix just by coating your plates with just simple ornithine. Someone was saying they have some doubt, so you can post it on the chat box. How many nitrogen in DETA silane? I do not know the structure of DETA silane, so maybe you can find it out and or maybe you can ask in the discussion forum. So you can just look at the structure and from there itself by looking at one monomer you can see how many nitrogens are there.